Okay, Bio30s, here we go with video 3 in our series on genetics. And, normally this would be like a really long lecture to you guys, a lesson in class, but I'm just going to have to split it up into a couple of short videos. And hopefully you guys are understanding this. Of course, you can always email me if you're struggling, but really do some questions and really do some practice. And so once we're done these videos, go ahead and attack the genetics notes and do some practice examples. The more you do, the better you get at it, and the more you'll have an understanding of how to do these genetics problems. So our last example, we had cystic fibrosis, and we had a probability of having a son. We said with cystic fibrosis, you can either have a son or a daughter. So the probability is 1/2. And then the probability of having an affected individual of cystic fibrosis, we multiply these together to get 1 in 8, or 0.125. So you guys have to be really careful when you're reading numerical response questions. Read the fine print. That's actually a really good life rule, guys, right? Is read the fine print. So here's an example. Let's say we just found this, and now they're asking you to record your answer as a percentage rounded to 1 decimal place. So how do we go from this decimal value to a percentage? Well, we times by 100%. And when we do that, we get 12.5%. So is that a percentage? Yes. Is it rounded to 1 decimal place? Yes. How would we write this in our little boxes on our numerical response answer sheet? Just like that. It's 12.5%. Now then, what if I had to record that answer as a value from 0 to 1 rounded to 2 decimal places? Well, I had 0.125. So I only have room for 2 spaces in my answer. So that means that this 5, this is just rounding, right? You guys have done rounding forever, right? The 5 makes the 2 round up, so it's going to be 0.13. So how do I write my answer? Well, 0.13, I write in 0.13, just like that. Is it 2 decimal places? Yes, 2 decimal places. How would I take this value and convert it into a whole number? Well, 12.5% is going to round up to 13. So as a whole number, it would be 13. Okay, so please pay attention to how you are going to express your answers. That's very important. That fine print is going to determine if your answer is acceptable and you get it right or not. So read carefully. Always read that fine details, the fine print. So how do you do genetics problems? You've probably clued into this already, right? I've talked about this a fair amount. Figure out what trait it is. Is it round or wrinkled? Is it tall or short for pea plants? Know what alleles. And then you set up a legend. Every question you should be writing a legend. What does big R mean? What does little r mean? Well, in our example here, big R is going to be round seeds, and it is dominant. And that means that little r is going to be recessive. So you're not using W for wrinkled seeds, right? You use the same letter. We know that round is going to be dominant to little r. So this greater than symbol means is dominant to. Okay, you do not use different letters. You use the same letter. You just use an uppercase as opposed to a lowercase. Okay, so the dominant is always the uppercase. The recessive allele is the lowercase. Then what are the parents? Well, first of all, the question may tell you what their phenotypes are. You have to figure their genotypes out. So if they're heterozygous for round, they're big R, little r. If they're wrinkled, then they are little r, little r. Then what are the gametes that they're going to make? So if the female is making eggs, or if the male is making sperm, you figure out the gametes, and then you put them out onto your Punnett square. So maybe this is the female parent here. She's recessive. She only has little r's, only recessive alleles. Here's the male parent. Well, he's heterozygous, so he has a big R and a little r, and we put them down the side of the Punnett square, and then you do your Punnett square. Set it up, and again, I feel that it is important that you actually write right in the square the phenotype as well. So this guy is round. and oh geez, I can't even spell today. This guy is round and this guy is wrinkled. Okay, and so you write inside each of your squares, is it round, is it wrinkled, what is the phenotype of that particular offspring. Then you'll be asked for the ratios, right? How many are round? How many are wrinkled? Okay, and so you have to figure out what are the ratios, and you may have to reduce to lowest terms. So if you look at this example here, I have two that are big R, little r, and there are two little r, little r. Is that lowest terms? No, it's not. I can divide 2 divided by 2 is 1. So for every one big R, little r, there's one little r, little r. Okay, so lowest terms is a 1-to-1 ratio for my, and this is a genotype ratio. These are the letters, the alleles together makes a genotype, not a phenotype. If I was doing my phenotype ratio, I would say there's one round pea plant for every one wrinkled pea plant. Okay, so make sure that you know the difference between genotypes and phenotypes. Okay, so next page, we are going to do an example together, and then I'm going to let you loose and do some practice. Then we'll come back and talk about a test crops. So next page, same legend that we just used. Set up your legend, big R for round seeds. They say a pea plant with round seeds is cross-pollinated. It's mated. They are putting together and mating two different pea plants. And so we have a round one mated with a wrinkled one. So first step, set up that legend. Big R, my capital letter is my dominant allele, and same letter, not W, but another R, but it's as small, a lowercase r, to indicate the recessive. Okay, so now what do I do next? Well, they have to now decide what is going to be my parent's genotype. So I have a round, and it's heterozygous times a wrinkle. So what are the letters going to be if it's heterozygous round? Well, it's going to be a big R and a little r. What about the wrinkled? Well, it's wrinkled because it has no big R's. It has two of the recessives. So there are the letters, the genotypes for these parents. Now what's my next step? Well, my next step is decide what are the gametes. So here's this heterozygous. Heterozygous has a dominant allele and a recessive. So the gametes produced by this parent are both of them. Wrinkled, there's no dominant alleles there, so it can only have recessive little r alleles. Okay, what is the next step that I would do? Well, my next step is to set up my Punnett square. So I take one parent and I put it across the top. So let's say this is the female parent, big R, little r. Here's the male parent, and now I fill in my Punnett square. So big R and a little r, big R, little r. What's that look like? What does this pea plant offspring look like? Well, it is going to be a round offspring. What about this guy here? It gets a little r from mom, sorry, a little r from dad and a little r from mom. He's little r, little r. Well, he's not going to be round. He's going to be a wrinkled offspring. How about this guy? Big R from mom, little r from dad. He is a round offspring. Last guy here, little r, another wrinkled recessive offspring. Okay, so there is our F1 generation. We have just set up our Punnett square. And now that we have our Punnett square, we have to figure out what is going to be our genotypes and our phenotypes. What ratio did we have of these offspring? So genotypes, let's look at the letters. Count up how many big R, little r's there are. There's two of them. And how many little r's are there? Well, there's two of them as well. Is that lowest terms? No, it's not. I can reduce that to a 1-to-1 ratio. What about phenotypes, the physical appearances? We'll count up. There's two round, two every, two wrinkled. Is that lowest terms? No, it's not. 1-to-1. One round to one wrinkled. Okay, so there we have our phenotype ratio and our genotype ratio. Okay, so next question then says, what about the F2 generation? We're going to take two round offspring from that F1 generation. Okay, so here is a round, and I'm going to mate it with another round. I'm going to cross-pollinate. So what offspring am I going to get in the next generation, the grandparents, the F2s? Well, let's take a look and see. You set up your parents, then you do your gametes, and then you put your gametes into your Punnett square, and you fill out your Punnett square, and let's see. Well, this should look familiar. When you have two heterozygous parents, you end up with a 3-to-1 phenotypic ratio. How many round offsprings do I have? Well, I have 3 round offsprings for every 1 wrinkled offspring. This is just like our pea plants when we did tall versus short. There was a 3-to-1 phenotype ratio. Why is that? Are all the genotypes the same? No, they most certainly are not, are they? When we look at these offspring in terms of the genotypes, I see there is one big R. These guys are both the same. There are 2 big R, little r, or heterozygous offspring. And then here's our one little r, and that guy has 2 recessive, so he's our homozygous recessive. Those are our genotypes. And how do we get 3? Well, these 3, 1 plus 2 is 3. That's our 3 round. And then where do we get our 1 white? Well, our 1 wrinkled? This guy here, this is our 1 wrinkled offspring because he has no dominant allele. He's got 2 recessives. Okay, so you guys are going to have a chance now to work through some monohybrid questions and do some genetics problems. And then I will be back with the next video in our series. Have at her, and we'll talk later.